So, in generally, we've had a lot of data coming up when it comes to vaccines per se. Because there's been such a huge number of people that have actually gotten vaccinations, we have a lot of data on safety, and it's one of the most safest drugs we've ever used. So, please, please, please be skeptical when you start reading things. Make sure that you see the data. It was just one stupid study that showed the link of vaccines with literally autism. But if you look at that, actually, it was actually the data was fitted to look like it was there. There was a very new study. I think it was Denmark or Norway. I think it was Denmark that they actually had a massive registry of millions of kids that had gotten the vaccine and had shown that there's no correlation at all. And this is a government entity. This is not a pharmaceutical entity trying to, let's say, force their own products on us. It was a governmental entity that showed that there's literally no correlation there. So please do not get discouraged from people like JFK Jr., who's just literally disgracing the whole scientific community. They're very protective. They're extremely protective against many different diseases, and we've eradicated diseases because of vaccines. They have a sustained protection, especially basically, they can really protect us quite a lot from against a lot of lethal diseases. And it's cheap. Vaccines are one of the most cheapest things because they don't cost that much at the beginning, and we only need to take them once, twice, or three times, basically for certain types. The thing about also that's interesting at vaccines is that they're not 100% effective, and basically how effective they are really differs among people. So you might, for example, let's imagine you have person A and person B, and they get the same vaccine. Let's imagine the COVID vaccine, and then you look at basically person A, they have much better antibody responses compared to person B. Why? There could be many different reasons for this. So vaccination is not 100% effective. However, the most important thing of vaccination is you develop something called herd immunity. This is when the majority of the population is immune. And what happens is that let's imagine you have 100 people. Within the 100 people, 90 of them have the vaccine and 10 do not have it. These anti-vaxxers, let's say. What happens with these 100? Technically, these 90 that are vaccinated, because they're vaccinated, the virus is not going to spread among them. So the virus is going to find it so hard to find a host that the 10 people that are there is very hard to them to reach it, because the virus will die eventually because it's not going to be able to find its host. This is herd immunity. This is why you need to have a certain amount of people be vaccinated against a certain against basically a pathogen that we're trying to protect ourselves from. And this is why it's basically vaccination is extremely effective. So let's talk about the types of vaccination. We have three main types. You have the whole organisms. This could be attenuated or inactivated. So these are you're literally giving the whole pathogen. Then you have subunit vaccines. These are literally pieces of that basically virus or bacteria that you're literally giving or vaccinating against. This could be capsular polysaccharides, inactivated exotoxins, or recombinant surface antigens. Then you have passive immunization, and then you have the novel forms of vaccines that are coming up that I really want to talk about today. So, whole vaccines is literally you're using the whole organism. So that's why we call them whole vaccines. So these could be live attenuated, or these could be inactivated. And when we say live attenuated, we basically make them in some sort of way crippled. So attenuated is when you cripple them. You make them not like barely able to actually really replicate. This could be, for example, with bacterial cells, or this could be with literally viral particles. So how do we attenuate them, or what does that mean when you attenuate them? You make them lose their ability to produce disease. So whatever bacteria or virus that you're producing, it will infect, but it will not cause the disease. Or it's basically you've made it lose its capacity to grow in a host. So especially the second one, the best way to do that is if you take any bacteria or any vaccine, any virus, and you make it grow in abnormal conditions. So what happens is that because it's growing in abnormal conditions, what happens is that they're going to mutate until they basically start to grow in those abnormal conditions. So what happens is that these new basically particles that are coming up that are mutated will not be able to anymore to grow in normal conditions. For example, BCG is literally a vaccine that is Mycobacterium bovis. So this bacteria, what we do is we literally make it live in a concentration of bile for 13 years. Literally for 13 years is living in that bile. So what happens is that this is basically starts to mutate, and then you have Mycobacterium bovis that's only able to live in bile. Then we take it, we inject it in our body. This Mycobacterium bovis is not able to live in our body anymore because it just got used to living in bile. We do this a lot with polioviruses. We put it basically in a monkey kidney epithelial cell. So we make it only used to basically replicate in monkey kidney cells. So when it comes to human cells, is not able to infect there. The MMR, so the measles, mumps, rubella are also attenuated vaccines, and the varicella are also vaccines like that. So we actually regard these as one of the best types of vaccines. But why are they so good? Well, purely is that they establish infection and extremely weak symptoms. So in generally, it's very safe. They confer lifelong immunity. So if you look at basically varicella, MMR, the polio, we only get it once. There's no boosters. There's no second vaccination or third or fourth. You only get it once, that shot, because it's so strong, the immune response that we get. Most probably because you're getting the whole virus or the whole bacteria to activate all different types of immune cell systems. So you get a overall good protection. You have basically a lot of different types of antigens. You're not just selecting one antigen to immunize. You can actually get an antigen or a response that against so many different types of proteins within the bacteria or viruses. And it's much more durable, the immunity, usually. So it's more cross-reactive. But there's issues. This is why not all the vaccines are live. So some of them, for example, the type 3 Sabine polio vaccine. So this is one of the type of polio vaccines. There's only literally 10 nucleotides out of 7,429 that are different from the wild type. So we took this polio vaccine. So we took this, literally the wild type polio that can cause disease. We put 10 nucleotides of mutations just so that it does not basically is not going to be able to cause disease. So that's why it's live attenuated. So you have attenuated, it's not able to cause disease anymore. However, there is a theoretical probability that it can revert back to its wild form, and that can cause basically death. For example, we also see this a lot with basically so we see risks with live vaccines with immunodeficient infants. So babies that are literally immunodeficient. So they're not able to really fight a good infection. So this live attenuated vaccine can sometimes, be a bit of a problem. And these live vaccines need to be in cold chain. They need to be literally at minus 80 degrees. So in certain areas, developed countries, that's not an issue. But in, let's say, underdeveloped areas like in Africa, where, having a good cold storage of minus 80 is not the most ideal thing ever. So you have issues there as well. So if we want to ask ourselves, live attenuated, are they the safest vaccines? Do they confer longest immunity? Do their immunity give us good B cells and T cells? Are they easy to store and transport? Well, no, yes, and no. They're not the safest. They are basically could give us the longest immunity, which involves both T and B and T cells, but they're not the easiest to store. So, because they need to be very in cold storage. So now let's talk about now inactivated vaccines. So these are basically, in some way we make these pathogens unable to completely replicate in the host. So we technically kill them. Either we kill them with heat, either we basically chemically inactivate them with formaldehyde or alkylating agents. Examples are the Salk polio and the pertussis vaccines. So, in generally, they're not as effective as live vaccines. And usually because they're not that basically effective, you usually need literally multiple doses. And in generally, you do not get T cell responses. You mostly get B cells. And this makes sense because these now bacteria or literally mostly viruses are not able to actually infect inside, so you do not have MHC1 presentation. So that's why with the inactivated viruses, you get B cell responses because they usually get eaten up, and because they're exogenous, they're on the outside, they're basically, will be presenting more the MHC2 pathway. The MHC1, they need to be alive and go inside. So that's an issue there. So, if you rank them again, they're safer than live vaccines. They do not give us the longest immunity, and usually it's because they only have B cell activation. Are they easier to transport? They're easier than live vaccines per se. So again, difference between attenuated and inactivated. Inactivated, you're literally killing them, either by chemical or radiation. Attenuated, you're just making them either not able to produce the disease or not able to replicate, but they're still alive. Well, basically as alive as they can be. So usually attenuated, you only get it once, while inactivated, you get it multiple times. Attenuated are less stable. They need to be at minus 80 degrees, while these ones are much more stable vaccines. Inactivated only produces B cell responses. Attenuated produces both B and T cells. So reversion from the virulent form. So is this vaccine attenuated? Can it go back to its pathogenic form, the one that causes disease? This could be possible. This is impossible almost, because you're completely killing that virus. So now let's basically go to a different type of vaccine, which is subunit. So this is where you do not use the whole pathogen, but you use basically parts of it. You take a part of it and you inject it, and that is your antigen. It cannot cause disease and is extremely safe. So it's one of the most safest ones, but they're not that effective. I'll talk of three main types of subunit vaccines. These are capsular polysaccharides, inactivated exotoxins, and recombinant surface antigens. So when it comes to the polysaccharide vaccines, so what we see specifically with Streptococcus pneumoniae is a very nasty bacteria that really lives basically within our throat. It actually is able to evade your immune system really well. Why? Because they have a polysaccharide capsule. So literally a capsule that is made of polysaccharides that kind of does not make your macrophages be able to recognize them and eat them up. So what happens is that what we can do is we can vaccinate against these kind of capsules. And technically we have in Streptococcus 323 different capsules. So we can take a bunch of these capsules and vaccinate against. But again, there's an issue because these are polysaccharides. Polysaccharides, antigens can be sugars, but they're not so effective. So you basically do not have a good stimulation of T helper cells. So you do not really get good CD8 T cells, and you get very poor antibody production. And you also have few to no memory cells. So that's an issue with polysaccharide vaccines. So let's imagine this is your polysaccharide. You do not get much very good memory B cell responses, not good T helper responses, or cytotoxic T cell responses. So one thing that we've seen that actually really helps really well is if we take this polysaccharide and we add a carrier protein. This carrier protein, basically, it could be something completely different towards, for example, Streptococcus. So let's imagine this polysaccharide is from Streptococcus. This carrier protein is something different. But for some reason, it just enhances much more, literally, your production of antibodies against the polysaccharide. So mostly because you get much more better CD4 T cell responses. So people have done it. Specifically, an example is against this Haemophilus influenza vaccine type B. So this is basically a vaccine against basically this type B capsular polysaccharide. So what we've done is we've literally added a tetanus toxoid. Something completely different. But this tetanus toxoid is a protein, and what it does, it literally just enhances more your CD4 T cells, which they can cross-react and make literally antibodies much better against the polysaccharide. So this conjugate vaccine makes it much more immunogenic, better antibody production, and much more basically memory responses. So now let's talk about exotoxins. So as we know, there's a lot of basically bacteria all around our basically the world and our systems that are able to produce a toxin. So a toxin that's able to cause literally damage. So it's not the bacteria per se that causes the damage. It's literally the toxin that it actually expresses that goes to our cell and directly kills it. These are, for example, is botulism, or basically tetanus. It's really these basically little tetanus bacteria, so these bacteria that produce tetanus that can actually cause harm. So what we can do is we can actually purify that exotoxin, like tetanus, and basically literally inactivate it using formaldehyde. So you obtain something called a toxoid. So it's a toxin that is killed, it's inactivated, because of course you do not want to, this exotoxin, you do not want to inject it, of course, because that's going to kill you. So you inactivate it and then you basically inject it. Once you inject it, you produce antibodies against that toxoid. So you now basically are able to protect yourself against the toxin. So we do this a lot for tetanus. And then we have recombinant and antigen vaccines. So what we do is if we have a protein from whatever pathogen, virus, bacteria, fungal, whatever, that we know we want to basically create an antibody response against, what we do is we take the genes that encode it and we create literally that basically that antigen. So hence it's recombinant antigen. We're creating it basically outside basically the body, that protein, and then we inject it. So an example for this is the hepatitis B vaccine. So we know in the hepatitis B vaccine they have the surface antigens. What we do is we take the genes that are actually encoding the surface antigen. We basically, in a system, basically with yeast cells, with eukaryotic cells in a cell lab, we create the protein, we produce a big bunch of it, and then we inject it in a human being. So this is what a recombinant antigen vaccine is. The next basically type of vaccination is passive. And passive immunization is technically, as the name says, it's passive. We literally are injecting specific antibodies purified from the blood of a donor. So this is sometimes, we usually have it when we do not have a very good vaccine in form, or if we have someone that doesn't have a good immune response. So we've even done it during COVID. So if you have someone that, let's say, recovered from COVID, has a good antibody response against COVID, you can take its serum that has the antibodies against COVID, and then you can inject it straight away into basically another person. And of course, that's going to protect it. But of course, you do not develop a memory response. So this actually is not basically, let's say, the most effective way of a vaccine. So we do this against a lot of different things like snake bites, botulism, rabies, hepatitis A and B, and tetanus. So usually, this is what you also use it a lot when you have been exposed to an infectious agent and you have no time to develop an antibody. Because again, developing antibodies can take about 2 weeks. So when you, for example, are bitten by a snake and that venomous toxin in your system can kill you within hours, you don't have time to vaccinate. You just give already antibodies that you know that's going to bind and protect you. But one thing, of course, I want to be also objective. One thing that of course, as safe as vaccines are, nothing in this world is 1000% locked in safe. Even we have some sort of misfortunes. An example for this is the H1N1 vaccine. So this is a influenza vaccine that has been associated with narcolepsy. Narcolepsy is a disease that what happens is that you literally could be running and then suddenly you literally pass out and you're just sleeping. So it's this kind of disease where you're not able to produce regulate your melanin. So you basically do not have control of your sleeping. So what they basically found out later on was that when you gave the H1 vaccine, you created a lot of CD4 T cell responses and CD8 T cell responses that were in some way, of course, kind of battling your H1 infection. But there was some similarity between the vaccine that we were injecting towards the H1N1, of course, but also certain sort of ion channels or certain, let's say, channels or ion channels in your neurons that were basically responsible for the melanin kind of regulation. So the CD4 T cells and CD8 T cells were basically kind of cross reacting. So not just killing infected cells with H1N1, but also any cell that had those kind of proteins, so these kind of ion channels. So you were getting narcolepsy. So, new vaccine strategies. Why do we need new vaccine strategies? Well, purely because we got outbreaks. This could be SARS, we could be Ebola, MERS, SARS-CoV-2, SARS-CoV-19. We need technically new ways to vaccinate basically against these pandemics that are sometimes occurring without us not knowing what they are. So we need platforms to actually be able to accommodate for these, pandemics that can occur, as we saw. And now I really got come up with basically COVID-19, because that was a very special case, and this is telling you this really opened up a new era for vaccination. So COVID, as you can see, COVID-19 really hit us badly, and the scientific community really rose to the occasion because we managed to get out a vaccine that was effective and very quick. So of course, these are just, if you guys are very interested, these are very good papers to read, along with these ones as well, specifically coming from Florian Kramer. Florian Kramer is one of the biggest people when it comes to vaccinology, specifically influenza, but he also worked a lot with COVID. He actually is a professor in Austria, but also in New York. He works at the Mount Sinai department. Met him, really, really nice guy. But let's go now to SARS. So we know in SARS-CoV-2, we definitely saw that in SARS-CoV-2 it has these beautiful, literally, these kind of corona, like crown-like trimers, kind of spike proteins that are able to bind to receptors, the host receptor, specifically ACE2. So this basically binding of this spike protein that we call S1 to the ACE2 receptor in the host kind of induced basically endocytosis. So of course, if you wanted to neutralize and prevent SARS-CoV-2 from entering host cells, we needed an antibody basically to come and bind to your spike. So that would basically give us some sort of protection from the SARS-CoV-2 from entering into our cells. So if we take now the S1 protein, so this is the protein that we exactly want to have an antibody response, we can actually even make it smaller, because what we see is that not the whole S1 protein binds to the ACE2 receptor. Is a very, very, very specific area that's called the RBD. That is literally the receptor binding domain. So it's the domain that's responsible for the receptor binding. So let's imagine this is the whole genome of your SARS-CoV-2. If we take just the S protein, it's literally almost 1,255 nucleotide, basically, kind of gene. And really, it's this part over here. There's literally RBD. So this is the binding domain, this is the RBM is the binding motif that we want to vaccinate and create an antibodies that are binding specifically to this area. Because if you block this area with an antibody, it's not going to be able to bind to ACE2, and it's not going to be able to actually induce endocytosis. So this is not the only, of course, antigen that we can actually vaccinate. We see that antibodies can also neutralize the nucleocapsid protein. Basically, we also saw that it can actually have other ones, maybe the envelope protein or the membrane proteins, but not really sure. But we also saw that, of course, if you have CD4 T cells or CD8 T cells, you can also combat SARS-CoV-2. And this can bind to almost quite a lot of different types of proteins within the SARS-CoV-2. But in generally, the main one was looking really at the spike protein, and specifically the RBD. So when we basically reported the first ever genetic sequence of SARS-CoV-2 was 11th of January 2020, extremely fast. Literally, we managed to sequence it within days that we knew that we basically were having this pandemic. So our DNA sequencing, or just genetically the genomic sequencing tools that we have, are extremely of high standard and up to pandemics levels. So we basically managed to get all of the sequence, and based on the sequence, we managed to actually get literally super quickly this RBD. Because what we can do is we can now use this basically kind of sequence to create either a subunit vaccine or basically a some sort of, let's say, recombinant protein vaccine, because we know what to produce. We know literally the DNA, the technically, let's say, the recipe to create that protein to vaccinate against. And if you would literally, for some people that have asked me, don't you think it was very fast, the production of the COVID vaccine? Yes, because traditional development of literally a vaccine can take 15 years or longer. This took 10 months. So extremely significantly faster. Why? Purely because the clinical trials were literally shortened. Also, basically, the FDA had a massive focus on it. So literally between the phase 1 and phase 2, there's literally a couple of years or a couple of months, basically like almost a year in between, between phase 1 and basically phase 2 and 3, because you're just waiting for the FDA to approve it. While here they were just approving it basically where as soon as the results came in, they looked at it and then they decided. Because here they have some time, and this time is that sometimes the FDA officers can just not give basically that much, can wait for literally months till they open the case. Well, here they were literally forced to. So, and generally, because we had a global pandemic and there was a lot of people dying, we basically had to make this shorter. So, and generally, this vaccine really saved us. And it really saved from a whole economic and also human, catastrophe of occurring. So now we knew that, okay, now we have exactly, we want to basically bind towards the S2, the spike protein. We wanted to basically specifically the receptor binding motif or domain. So what are the strategies that we can use? We could use the weakened virus, so we can take SARS-CoV-2, make it weaker, attenuate it, and basically this would get your immune response. But generally, this never really worked. Either you get a weakened virus or inactivated, wasn't the best. Then you can either do the protein-based vaccine. This is basically the recombinant protein, because now we know exactly the sequence of the spike protein. You can create it, and you can basically inject it. Not the best. Then you can actually use a viral vector. These were the new vaccines that came out. So what you can do is you can use literally a virus like adenoviruses that are very stable. You can make them very safe, not cause any disease. And what you can do is you can genetically modify this adenovirus to also express the COVID spike gene. So what happens is that when you inject it, it literally is going to infect normal cells and express the spike protein. This spike protein then can be taken up by literally your dendritic cell and actually induce an immune response. These are extremely really good vaccines that actually worked, and these were actually your Johnson & Johnson vaccine, your basically AstraZeneca vaccine. So these were actually really decent vaccines that were actually all around. But the really one that took the spotlight, I have to say, are RNA vaccines. So what are RNA vaccines? Are literally a piece of mRNA that actually encodes to whatever protein or antigen that you want to vaccinate. Because we knew literally the spike sequence, we could actually use an mRNA. But this mRNA is special because